This? Okay. Hello, my name is Cassie Garza, and this is going to be my seminar presentation over how DDT impairs lectin development in lymphoblasts. So this image right here, it just shows a cell responding to these lectin proteins, which we're going to go into later, and how they can stimulate mitosis in cells. So that's a major theme of this project. So the discovery of dichlorodiphenyl trichloroethane, otherwise known as DDT. So DDT was first synthesized in 1874 by a chemist named Othmar Zelder. So it was not a naturally occurring substance. It did have to be synthesized in a lab. And then in 1939, wasn't until that time period later that another chemist, Paul Mueller, discovered the success of DDT as an organochlorine pesticide. So it was actually one of the first, it was the first organochlorine pesticide that whose use became popular in about the 1940s. And DDT is actually accredited with a lot of major contributions. So like during World War II, it was used to control typhus and other kind of mosquito-borne illnesses. It controlled crop pests, which provided a food surplus worldwide, kind of allowed the population to grow. And it's also credited with the near eradication of malaria in several different countries. So just a specific incidence of that, like in India, it brought the cases of malaria from about 100 million to only 100,000 cases between the 1950s and 60s. There's a huge difference there, and that actually made DDT the most effective preventer of malaria to date. So we have nothing else that we've tried has worked as efficiently for that as DDT. So the chemical structure of DDT and the metabolism to DDE. So the chemical structure, DDT is what's known as a polychlorinated biphenyl structure. So that just describes polychlorinated, right? That you have multiple chlorine groups, like all of these on the outside. And biphenyl, just phenyl groups are going to be these rings here. So biphenyl, that it has two of those. So these phenyl groups are made out of carbon and hydrogen bonds in a ring, an aromatic ring. So that actually makes the compound very hydrophobic. So that means that it's going to fear water, and the compound is going to dissolve readily in lipids or fats. So that's just characteristic of these rings here. The other major part of this chemical structure is going to be these chlorine groups. So chlorine as the atom is actually a halogen. So that means that it's going to have 7 outer electrons. So it's a very reactive just molecule. It's trying to get its stable 8 electron configuration. So all of these chlorine groups give it a high reactivity, and these biphenyl groups are going to give it the hydrophobicity that's characteristic of the chemicals. So DDE is the single major metabolic metabolite. There you go. It's going to be the single major metabolite of DDT. So with this image here, you can see exactly what's missing. You're missing a chlorine group here on the end, and that'll create a double bond here. So you're also end up missing a hydrogen atom from here. So it shows the total atoms that you end up missing are going to be like a hydrochloric acid molecule. Anyway, DDT is just a major metabolite of DDE is a major metabolite of DDT, as I mentioned. In the body, it's converted by the protein cytochrome P450. So this protein is actually very popular in the metabolism of many drugs in the body. Like I said, these molecules, they're very hydrophobic. So another way to say this is that they have a very high partition coefficient. This is just an advanced measure of a chemical solubility in lipids. So just another way to say that. So this lipid solubility, it leads to the accumulation of these chemicals in the fatty tissues of bodies. So this is going to come up in the next slide or two, how it accumulates in organisms and just that it stays there over a long time. So it would have a long half-life. So we're going to move on to the next slide. Where DDT accumulated in and damaged the environment. So I just stated that DDT and DDE are hydrophobic molecules that accumulate in lipids. So this property has led to the massive accumulation of DDT and its metabolites in the environment. Back in like the 40s when they started the use, and the few decades after that, the use was very popular. Like I said, it had a lot of positive impacts. So the use was very popular, but it was accumulating in the environment. And one of the first people to see this was a marine biologist and conservationist known as Rachel Carson. So she wrote her book, Silent Spring, in 1962, which warned society about the dangers of DDT. So she followed cases of animal deaths resulting from DDT spraying. She gathered research data, and she came to the conclusion that DDT can cause cancer, and that it was just a threat to the environment because of its accumulation. The book was actually very controversial because, like I said, it was doing great things around the world, like eradicating malaria, which was unheard of at the time. So she kind of got a lot of hate for that, but she was indeed right. It was actually having some really profound effects on the environment. So DDT was eventually banned in 1972 in the United States in an effort for environmental protection. There's actually research in 2018 that took place in European countries, but it found that 90% of mothers and their children have detectable levels of DDT and its metabolites in blood and urine samples. And this is even decades after DDT was banned in those countries. So that just shows you, that it really does accumulate in the fatty tissue of organisms, and it does have a long half-life, so it will stay there for decades after. So as I mentioned, DDT affected many organisms. It was particularly detrimental to these guys. This is the bald eagle. So it actually brought the bald eagle population in the United States down to almost extinction in about the 1960s. So what it did was it interfered with something called the calcium ATPase pump. So this was essential for the correct development of the eggshells of, I guess when they lay their eggs, the calcium ATPase is essential for creating a strong eggshell, just a properly functioning eggshell. And DDT interfered with this process. So what happened is, for generations, it was very hard for any bald eagles to survive, even just the egg hatching. And so, yeah, we almost lost all the bald eagles in America just thanks to DDT. So we're going to move on to our next slide. DDT is an endocrine disrupting chemical. So the endocrine system is a network of glands all over the body that maintain homeostasis. They kind of keep the body going using chemical messengers known as hormones. So hormones can either be amino acid or protein based, or they can be steroid or lipid based. And they're going to bind to receptors on the cell, kind of like we see here, right? But we'll get into that later. They'll bind to receptors on the cell or organs, and they cause a generally slow but prolonged effects over the body, just like I said, to control homeostasis. Endocrine disrupting chemicals are chemicals that interfere with the cellular pathways and function of the endocrine system. So this can include interference with the synthesis, with the secretion, transport, binding, even the elimination of hormones by endocrine organs. So they can function by either mimicking these hormones, they can inhibit these hormones, or they can decrease sensitivity of the cells or organs to these hormones. So this image here, as you can see, it's about endocrine disruptors. So you can see these structures over here are going to be what the normal hormone is supposed to look like. And these kind of W-shaped structures are going to be endocrine disruptors. So here it shows you just the way that it's supposed to work. The hormone binds to the receptor site and then would cause its cell response. But these endocrine disrupting chemicals are going to inhibit. This one is acting through inhibition. So this one is just going to bind to this site, and it doesn't cause any physical response itself. But as you can see here, it is going to be able to block the hormone from causing its response. So in that way, it does inhibit the hormone. So there has been numerous studies on rats that have shown DDT to be particularly devastating to the reproductive system. So DDT affects these sex hormones mostly because these sex hormones, they're going to be those steroid or lipid-based hormones that I discussed, right? And like I said, DDT dissolves in lipids. So it's natural that that would be the hormones that they really affect the most. The results of these studies yielded similar conclusions. So some of these include like a decreased penis size, decreased sperm count, sperm motility, disruption to the menstrual cycle, and even disrupting like follicle release in females. So yeah, this was just data from experiments with rats. And then there's also another study that occurred in Florida. It explored the effects of DDT on the local alligator populations after DDT spraying for insects. And it showed like the same results, even in nature with these alligators, same kind of reproductive system related results. So the thymus is an organ of the lymphatic system. So the lymphatic system is a network of vessels that kind of run throughout the body. And what it does is it collects extracellular fluid from the tissues, and it also houses cells that are crucial for the immune response. So it's going to be located behind the sternum, like in this picture here. And its size will decrease after puberty because most of the cells should be developed around that time. So after that, into adulthood, it shouldn't be doing as much development, so it is going to shrink. So it houses immune cells, like I had said. More specifically, these are going to be known as thymic lymphocytes. So the thymic lymphocytes, otherwise known as T cells, they're going to be referred to as both interchangeably. They are just two names for the same structure, T cells or thymic lymphocytes. So they're going to mature in the thymus through a process of differentiation, where they'll kind of become unique to their own function. They develop a function and receptors for that function. So yeah, they'll develop there in the thymus before traveling to the body, to the lymph nodes, to the lymphatic system. Like I said, this function should happen from before birth, when the cells are made, for lack of a better word, to the time of puberty, where they really get to develop there in the thymus. So the subcapsular layer is an important structure to know about the thymus for some of the results for later. So the subcapsular layer is just kind of going to be like, if we're going to look at this one, it would kind of be on the outside. It's just going to be a subcapsular layer of what are called lymphoblasts. So those are just going to be like new cells. Those are the ones that are replicating through mitosis just to create more cells. So that's what we need to know about the thymus for our data that's going to be presented later. So the thymus is divided into two regions, those being the medulla and the cortex. So there's other structures in the body that are divided similarly, but here in this picture, you can see the cortex is going to be this outside region, and the medulla is going to be all of this inside region. So the medulla is home to these structures known as Hassel's corpuscles or thymic corpuscles, same structure. So their function is, as relevant to this presentation, at least, is going to be the programming or the maintenance of regulatory T cells, which those are going to regulate the actions of other types of T cells. The important things for you to know about corpuscles is, like I just said, they program regulatory T cells, and also that they develop in 3 different stages. So these 3 different stages, the third stage is going to be corpuscles that are fully developed and they have full function. The second stage is going to be they're partially developed, they have partial function, and then the first stage is going to be they're totally underdeveloped and they don't really have function at that point. So a lot of cells being in the first stage is not really good. That would mean that there's less regulatory T cells, which could lead to diminished immune responses. So the stages of these corpuscles, they can actually be analyzed under a microscope. So we're going to get to see that later. All right. So thymic lymphocytes, or T cells, right, interchangeable, are a type of white blood cell that fight off invading pathogens in the body. So there's a few different types that we haven't discussed yet, but the important ones for this slide are going to be the cytotoxic T cells that destroy infected cells. They actively destroy them. And helper T cells, which send signals that direct other immune cells to come and fight the infection. So T cells recognize antigens that are present on the outside of infectious agents. So they're unique to these specific antigens, and they'll respond to those antigens. If a pathogen comes into the body with antigens that the body has already seen before, then the appropriate T cell will respond, it'll bind to it, and then it will make copies of itself and it'll destroy the pathogen that way. In the case of novel pathogens that come into the body, Memory T cells will help create memory of the receptors that are on the pathogen. And that way the body will be able to recognize it and respond to it in case of reinvasion. So this makes T cells a part of the adaptive or acquired immune system, thanks to the function of these memory cells. So in this image, you can see it shows these little blue balls over here are going to be T cells or thymic lymphocytes, right? And it shows them coming from the bone marrow. So I don't know if I failed to mention this earlier, but like I said, when they form prenatally or before birth, they actually form here in the bone marrows where these cells are initially created. And then kind of around the time of birth or right before, they will migrate to the thymus. Like I said, where they'll undergo development and differentiation. So that's exactly what you see in this picture here. You can see these T cells developing all of these receptors on their surfaces. And then you can see over here where they turn into mature T cells. They have two very important, these are very important, and we're going to see them in the next slide. So you can see this one that says CD4 is actually going to be it has the proper surface receptors to be a cytotoxic T cell. And the CD8 has the proper receptors to be a helper T cell. So we'll get to see that in the next slide in a little bit more detail. So in this slide, we're going to describe how T cells recognize intruding pathogens. So the first step is going to be there are these antigen presenting cells that will locate the intruder, and they're going to attach a major histocompatibility antigen complex. So they will attach these to the intruder, and that structure, that MHC structure itself, is what the T cells are able to attach onto. So that's actually where they're going to bind. It's going to be to the MHC antigen complexes. So there's a few different variations of that complex. The ones important to this study are definitely going to be the MHC1 and the MHC2. So MHC1 is going to bind to the CD8 receptor, like we saw earlier, like in this picture, like these CD8 receptors. So the MHC1 is going to bind to the CD8 receptors, which is going to program for cytotoxic T cells, or it's characteristic for those. And the MHC2 complex is going to bind to the CD4 receptors for the helper T cells. So yeah, the type of MHC complex is going to determine what type of T cell is going to develop. So as you can see here, this was the clonal expansion. This is just depending on what type of MHC complex here that it's exposed to, it can develop to be either a cytotoxic, memory, suppressor, or helper T cell. So let's see if I missed anything here. Yeah, that's about it. So you will get the T cell depending on what MHC antigen complex is, which again is attached by these antigen presenting cells. And those MHC complexes are what the T cells are actually able to bind to. And then so T cells are only activated if or once they bind to their matching receptors. If they fail to bind, then there's not going to be any reaction, and that's just to ensure that you have the right T cells with the right pathogens interacting. And so next. So here we're getting to the lectins. The lectins are a type of protein that bind to specific carbohydrate receptors on cell surfaces. So lectins are naturally occurring in nature. They're in virtually all of the plant foods that we consume. They're especially present in beans, wheat, peanuts, things like that. They have been linked with health conditions like diabetes, autoimmune diseases, just kind of a side note. So lectins participate in the immune system by binding to carbohydrate receptors on cell surfaces, and they can thus carry out a variety of reactions in the body, which is why I picked this lovely image here. So it shows some signaling pathways at the body. So like this one here all the way on the left with these kind of green lectins, that's going to kind of signify like typical cell signals. So to me, this is kind of similar to like how amino acid hormones would bind. They'll kind of bind to the outside here, and then that will subsequently cause a cascade of reactions inside the cell. But that's just signaling inside the cell, and then whatever function should occur will occur thereafter. So the more important one for us is going to be definitely these guys. So these are little pathogens, and you can see all of these things sticking out on their surface are actually going to be lectins. So The lectins are going to bind to carbohydrates on the cell surface. And yeah, that's just recognition of the antigen. So the specific lectins will bind to the specific receptors. And here, this image even takes it further. It shows the pathogen being taken into the cell, the process of endocytosis. It'll grab it with a vesicle and just pull it into the cell. If we look over here, you can see these colored structures, multicolored structures on the outside of the cell. So those are actually going to be like carbohydrate receptors or glycoprotein receptors. Those are like receptors that would bind to lectins. And this just shows them being made inside of the cell. And then once they're developed, they'll join the cell surface and become active receptors. And then the last one, we can see lectins here. They're bound to the receptor on the cell, but then they're also bound to each other. So that's going to help in cell adhesion, which is keeping cells in close contact with each other. So those are just some of the many functions of lectins. As I mentioned, this one here, the pathogen recognition, is definitely going to be the most important for us to understand for this presentation. So concanavalin A, or ConA for short, is just one example of a lectin that's going to be used in the experiments later. So ConA binds to and activates T cells and causes an anti-cancer immune response. So ConA is also known as T cell mitogen. Mitogen just refers to a protein that induces cell division. So ConA would be considered mitogen. And it acts directly with like tumoricidal and immunogenic effects. So ConA will induce a healthy and normal rate of cell division if it detects cancer cells or like cells that are abnormally dividing. It does have the ability to activate T cells and direct them towards these cancerous cells to fight them off and try to eradicate them before cancer actually develops. So here in this illustration, we see ConA, the lectin, is going to be this light blue molecule here on the left. And you can see it kind of binding between these surface glycoproteins and the T lymphocytes. So ConA can bind glycoproteins and commit T cells to proliferation. So that's what ConA is doing over here. And then here on the right side, you can see it interacting in a similar way, but it's going to be with the MHC2 complex. So if we remember that, that'll go to the helper T cells. I don't know if it's really relevant for this picture. You can see it interacting here. A Kuffner cell is just a type of liver cell. But what's interesting is that ConA here detected something abnormal with this cell. So it relayed this message to the T lymphocyte, and it produced these TNF alpha factors. So those are tumor necrosis factor alpha, which is just a cytokine factor that's seen during like pathogen-related inflammation. And it's also seen in cases regarding apoptosis. So apoptosis, you can see the word here, that's just a programmed cell death. So the cell intentionally kills off these cells because it senses something wrong with them. It realizes that they're not correct. They're not good for the body. So you can see ConA here working with the TNF alpha factor to induce apoptosis, which is going to stop the proliferation of this cell. You can see there. And yeah, it'll stop the proliferation of this cell by signaling the abnormality of the cell to the T cells, which will then initiate apoptosis. So we have Ki67 on this slide, which is going to be an antigen that's encoded by the MKI67 gene. This region is a protein coding region that's heavily associated with the cell cycle and cell proliferation. So it's a great biomarker for cell proliferation since its presence increases with the level of proliferation of the cells. So it makes it an excellent marker for diagnosing cancer. It's really popular for breast cancer at the moment. They're kind of exploring the uses to diagnose other types of cancers, but it's really useful for breast cancers. Okay. So if we look at this image, we can kind of see how it works. So just the main point is that when cells are proliferating, they produce this biomarker Ki67. So it's not present in normal cells. If you see just mature cells that are not undergoing mitosis, you won't see Ki67. It's only present during mitosis. So I really like this image. You can see the cell proliferation, and then it's going to lead to the presence of the antigen Ki67, which has some uses that some of we've already discussed. Like I said, it's a proliferation marker. It shows the presence and the amount of cell division that's going on. It can be used as a diagnostic tool, as I mentioned with breast cancer. It's used to diagnose breast cancer. There also can be a prognostic indicator. So it can do this because, like I said, the higher the level of cell division, the higher the amount of Ki67. So if you take the measurement and you analyze it and it's rather low, there's just a fairly low amount of Ki67, you can correlate that with kind of a slow growth tumor, or just slow mitotic rate, versus if you see extremely high levels of Ki67, it's probably a very fast growing tumor. There's very rapid mitosis undergoing. So that can kind of help you with the prognosis of a patient. Like that already has cancer, you can use it to give them a prognosis, kind of see how fast that tumor or those cancerous cells are dividing. Okay, so with that, we're going to move on to our hypothesis that consists of all the background information that we're going to need. The hypothesis here is going to be that DDT exposure inhibits lymphoblast differentiation, leading to a decrease in functional leptin receptors and a diminished response to concanavalin A. So let's get straight into the results. So this is the first of 3 slides, 3 consecutive slides that are going to be from the same experiment. So in this experiment, they took pregnant female rat dams, or just pregnant mothers, and they were exposed to a water-soluble isomer DDT while they were pregnant, and then also while they were lactating. And they kind of did this, the scientists in this experiment did this to reach a certain level that is actually found in humans. It's called like a low dose exposure to DDT level that is found in humans. So they try to use that same level to give a better comparison. So the sons of the mother rats that were exposed, those were the actual subjects. So it's more like a prenatal disposal that they received. So yeah, what they did was they excised the thymuses of the male rat offsprings, and those were analyzed on the 7th, 42nd, and the 70th days. So those correspond to the 7th day is going to be like a neonatal period, the 42nd is going to be puberty or adolescence period, and the 70th day is going to be adulthood. So like I said, the male offspring of the mothers that were fed DDT are going to be what were tested, and then the controls are going to be male offsprings of mothers that were not given any DDT exposure. Also analyzed at the same age. They also had their thymuses excised and analyzed at the same age. So for this particular slide, the technique used to obtain the results was the proliferation assay test. So an assay test is just an analytical procedure that's commonly used in the laboratory. The technique allows for the evaluation of cell proliferation activity that's really close in accuracy to the real proliferation rate in vivo. So how they did this was they added a radioactive nucleoside, 3H-thymidine, to cell suspension samples, and then they incubated these for about 4 hours. And then the cells were collected and counted by a cell harvester. So if you look here, you can see on the x-axis, like I described, was these days of postnatal development. Once again, neonatal, puberty, and adulthood. And then the y-axis for this CPM is going to be cells per minute that were counted by the harvester. So the pattern of proliferation here is really interesting. If we look here at the 7th day, you can see the control rats had a significantly greater value of proliferation rate than the DDT exposed rats, but both values are fairly high. Okay, you can see that both are fairly high. And then moving on to the 42nd day, you can see the control decreased quite a fair amount, while the DDT exposed samples barely decreased. They barely showed any decrease at that rate. So if we move on to the 70th day, you can see a sharp decline in the control samples, where their proliferation rate of the T cells greatly dropped by the 70th day, which is the adulthood marker, right? But if we look at the DDT cells, they actually remained quite elevated. They did decrease a little bit, but they're definitely beyond the level that it should be. So, so the results that the proliferation of thymic lymphocytes in DDT exposed rats showed barely any decrease from the neonatal to the adulthood, like I said. And the implications of this, well, you can kind of see. So I want to discuss why the control would decrease like that. It makes sense, like I had mentioned, that the cells should be differentiated and properly functioning by about the time of adolescence. That's why the thymus is going to shrink after that. The cells should pretty much be done developing. So it makes sense. It's natural that you would see the development of just a very low proliferation rate, right? Just for maintenance. So the fact that DDT remained very elevated, the implications of this are going to be that DDT does interfere with the normal proliferation rate of T cells. And the importance of this is really going to be seen in the next slide. But of course, you can visualize that this would just mean a lot more extra lymphocytes. But you're going to see just kind of the specific kind of lymphocyte that's being formed in the next set of results. So this data was gathered from the immunohistochemical evaluation of Ki-67. So the immunochemical evaluation is a process used to assess the presence of a specific antigen by using their specific antibodies. So they'll bind to each other, and that reaction can then be visualized under a microscope. So the width of the subcapsular layer of lymphoblast is a great index of overall proliferation rate at the thymus. It's just a great indication of that, and an increased width of this layer is going to be correspondent to just an increase of lymphoblasts, which we'll see in this slide and the next slide after. So the x-axis, if you look here, is going to be the same. You have the 70th, 42nd, and 70th days of development. The y-axis here is going to be the measurement in micrometers of the width of the subcapsular layer of the excised thymuses. So if we look at the 7th day, the test subjects had a slightly greater width of the subcapsular layer, but the difference there between that one and the control is not going to be statistically different yet. But they are both quite high there. And then you can see that moving on to the 42nd day, there is also no statistical significant difference there, but you can see that both values did decrease going into that 42nd day. And looking at the 70th day, you see the values stay nearly the same, but the control value does decrease enough to give the difference here at the 70th day a statistically significant difference. So, yes, you can kind of see it. Once again, it makes sense that these values should decrease. You shouldn't have a really thick subcapsular layer, but DDT exposure did create abnormalities here as well. So as mentioned, just the greater width of the subcapsular layer does mean more lymphoblasts. So lymphoblasts, as we discussed, are like the new cells. They're the ones that are replicating. So they're underdeveloped. They haven't undergone differentiation. So that means they're not gonna have all of the lectin receptors. They may not all be developed, and this can lead to the diminished responses that we're gonna see to lectins later. So the last data results showed us that there is an increase in lymphocyte proliferation, right? But this result gives us more information as to why this is occurring. So it's going to be occurring because this lymphoblast layer is enlarged, and we're having a greater number of lymphoblasts. So the last results kind of let us know that there's more lymphocytes in general. This one lets us know that it's really that there's more lymphoblasts. So really it's more that there's more underdeveloped thymic lymphocytes going on here due to the DDT exposure. Well, moving on to the next one. This is actually just the images from the last slide. Just the microscopic images of that, of the immunohistochemical evaluation of the Ki-67. So A here is going to represent the control group, and B is going to represent the DDT exposed group. So the black cells are going to be lymphoblasts. So like I said, Ki-67 is present in cells that are mitotically dividing. So that's going to be the lymphoblasts, of course. And then these blue cells are just going to be, we can think of them as T cells, regular T cells. They're adult, they're already matured. They're not lymphoblasts, so they're more developed than that. So we're going to look here at the control sample. The first thing that we're going to look at is this kind of border edge. So this is the subcapsular layer that we were talking about in the last slide. You can see that in the control, it's rather thin, and it's kind of got that line here on the outside. That's where the layer should be, that subcapsular layer. And then you see a few here within, but that's normal. There's a lot more lymphocytes than there are lymphoblasts. So like I said, that's our control. That's going to be normal, what we expect, what should be happening there in the thymus. Here with the DDT exposure, first of all, you can see that the subcapsular layer is significantly thicker. It's probably at least twice the size of this control layer. And then also looking deeper into the thymus, you see that there's also a lot of lymphoblasts that are occurring there. There seems to be almost as many lymphoblasts as there is lymphocytes, which is definitely not a good sign. Means that there's far too many immature lymphoblasts in the thymus. And like we've discussed, these lymphoblasts are going to be underdeveloped, so they'll lack lectin receptors. And that statement right there that I just made, it's going to be further supported by the results regarding concatenated A that are going to come up in a little bit. But yeah, so this is the depiction of Ki-67 and the abnormalities that we see there. So this is from a second experiment that was performed in a very similar manner to the other one. So it also used male Wistar rats as the test subjects. It also fed them DDT to mothers through their water. And then it also analyzed the excised thymuses of the male progeny. So it was a very similar experimental design. I believe it was done mostly by the same scientist. But it did cover something entirely different. This research was really looking into thymic involution, which is just the shrinking of the thymus that normally occurs with age. But this particular part of the experiment is really just looking at the thymic corpuscles and what happened, what changes in those DDT caused. So here in figure A and B, there are images of thymic corpuscles from the thymus, from the thymic medulla. So you can see that the control sample, you can really only see one thymic corpuscle that's present. And then in B, the DDT exposed sample, you can see 1, 2, and a third one here that are present. So there's definitely more thymic corpuscles going on. You can see that. And graph C actually shows the amount of thymic corpuscles per millimeter squared in both of these samples. And you can see that this green bar, the DDT exposed, is about twice as high as the control value. And then figure D over here actually breaks it down even further. So D is going to show us exactly how many of these corpuscles are in what stage of development. You can see that the DDT exposed has many more that are in the first stage of development, and then it has many less that are in the second and third stages of development. So remember I said that first stage is going to be very underdeveloped cells. And so that's just kind of correlating with everything that we've been saying. So more underdeveloped, DDT is causing more underdeveloped cells. And these cells, they're gonna have consequences based on that being underdeveloped. So these particularly, remember the thymic corpuscles, they modulate regulatory T cells. So an increase in the number of immature corpuscles negatively impacts the control of T cells, which is also playing a role in their lack of response to lectins, which we're about to see in more detail with this next slide. So the results here are also from that first experiment that I detailed at the very beginning of the results slides. For this part, 5 micrograms per milliliter of concatenated A solution was added to control samples and to test samples. And then the cultures were incubated for 72 hours at room temperature. And then not at room temperature, at 37 degrees Celsius at the body temperature. And then the same nucleoside, the 3H thymidine, is going to be added here. And the cells were also harvested and counted in a similar manner to that other data. So here you have the same x-axis, of course, the 7th, 42nd, and 70th days of development. And then here on the left side is going to be an index of proliferation, which is just the measurement of that 3H thymidine nucleoside, the measurement of that nucleoside's incorporation with con A in the lymphocytes was measured to give us an index of proliferation. So at the 7th day here, we can see that the proliferation rates of the DDT sample are just slightly higher than the controls, but there's no statistical difference there. Moving on to the 42nd day, we can already see a huge jump in this. You see that the control actually increased some, and the DDT exposed samples decreased significantly, probably almost by half there, it appears. And then moving into the 70th day, both values decrease, which end up leaving the controls with a very high value and the DDT exposed thymuses with a very low value. So these values are regarding, right, the response to con A. So response to the lectin con A, remember that's, remember what that is? It's going to be like the proliferation response of T cells as an anti-cancer immune response. So you can see that the control would have the normal values of response to lectins, and it should be at this level. And DDT has diminished that to almost half. So you have basically half of the response to concanavalin A, which is definitely going to have some implications. It's definitely going to have some implications with immune responses, like I said. So we have seen already that DDT causes high amounts of immature and underdeveloped lymphoblasts, but this data here really helps us solidify the lack of functional lectin receptors that correspond with this. So we know that there's a lot of lymphoblasts, and this testing with con A is showing us the consequence of that, which is going to be a diminished response to lectins, which would occur according to my hypothesis, by a lack of functional receptors, which, when cells don't have the time to differentiate and develop like they should in the thymus, they're probably lacking these receptors, and that's what's causing this diminished response to lectin concanavalin A. So with that, we're going to move on to our last results slide. So this last piece of data is from another different research article, and it's here to provide that last missing piece of evidence to kind of tie together my hypothesis. So the data presented here, much of it is about factors that are kind of unimportant to our study. So we're going to disregard those. The only really relevant part of this one is going to be this value, the T-independent antibody response in mice. So antibody responses are the result of the attachment of lectins, right, on an antigen to lectin receptors on T cells. That's how you're going to get an antibody response. There's no other way around that. So this is definitely going to show at what age lectin receptors in mice are at the adequate level, the level that will provide a proper immune response. So this research determined that age. Remember, it's like the age of full lectin receptors and full antibody response to occur in mice between 41 and 56 days. So we're going to use that data and kind of go back to this other slide. And what we're going to look at is this middle value, right, the 42nd day, because that's the value that's going to fall within those days. So according to this research, right, mice, rodents should have the complete proper antibody response levels by the 42nd day. So if we look at this one and we look at the control, this value here would represent the normal immune response value that is attained by the complete T cells that have full lectin development. So as you can see, the DDT exposed subjects, they have almost half of this. And then even going into adulthood, they retain levels that are less than half of what the proper level should be. So the implications of this, it's just gonna keep supporting my hypothesis. So since DDT affected T cells never attain the response that these fully developed control T cells attain, it supports my hypothesis that DDT inhibition of lymphoblast differentiation leads to underdeveloped T cells that lack functional lectin receptors capable of normal antibody response levels. So with that, we're going to move on to our conclusions. So DDT exposure causes the following changes in the thymus. It changes the proliferation rate of thymic lymphocytes and causes them to remain elevated beyond the control rates, causes an enlarged subcapsular layer, which produce an elevated number of underdeveloped lymphoblasts. It increases immature thymic corpuscles, which produce immature thymic lymphocytes or lymphoblasts. And it causes thymic lymphocytes to have a greatly diminished response to lectin concanavaline A, which will remain greatly below the normal T cell antibody response level. So overall, all of this data does validate my hypothesis that DDT exposure inhibits lymphoblast differentiation, leading to a decrease in functional lectin receptors and a diminished response to concanavaline A. However, novel research about certain areas like the developmental process of lectin development in T cells, as well as like what age this occurs, novel research in those areas could definitely provide some more solid pieces of evidence to strengthen my hypothesis. But as of now, it is validated with the results that I have displayed. So our last slide, of course, is just going to be our references. And thank you very much for listening to my presentation.